Righto, so in today's video that we'll be covering for Year 11 Bio, we're going to really be continuing on with the whole cells stuff, and really we're going to focus on looking at cells and the specifics of what's inside of cells. And these specifics we call the ultrastructure, alright? So by the end of this lesson, and hopefully by the end of the prac we're going to be running on Tuesday, you're not going to have this issue where what you can see under a microscope in biology is black, 3/4 is black, then most of that last quarter is your eyelash, and then a tiny little fraction is actually just the actual slide. Hopefully we're going to get at least a nice 3/4 of the actual slide, but sometimes microscopes can be a bit tough. We're still in Nelson Biology page 6 to 18. We haven't really gotten off that, but it's just a bit more specifics on those topics. So in the study design, we're looking at the ultrastructure of plant and animal cells, okay, in terms of their organelles, and identification of these organelles using both the light microscope, which we have and will be using in class, but also the electron micrograph, which is a little bit more high-tech. So what is a light microscope and electron micrograph? Hopefully a light microscope looks pretty much the exact same as what you're expected to see and you know and you've been using quite a bit in class. But this is an electron micrograph, which most of you probably haven't seen. So a light microscope is actually really simple. You pretty much have this illuminator which shoots light up through the stage, right? And you'll have a cover slide here on the stage with whatever you want to look at, which you're interested in, and that light goes through that cover slide, through all these series of lenses which magnify the image, and then into your eye, which is going to be over here. All right. Pretty much the light that doesn't get to your eye makes out as whatever the specific thing that you're looking at is. Right. Electron micrograph, on the other hand, works in a little bit more of a complicated way. You've got electrons actually, so you'll have the whatever you're of interest in this little stage area here, but you actually shoot electrons at whatever you're interested in. Those electrons bounce back in different directions, pretty much, and all of this fancy computer software picks up those electrons, and then you'll get the image on our computer. All right. We can get color out of the light microscope. We do not get color out of the electron micrograph. All right, but we get maybe 400 times magnification here. Here we can, here with the electron micrograph, we can actually see atoms, which is incredible, something we haven't heard of in the past decades or so. Alright, so hopefully you know now that this is a eukaryotic cell, and it's more specifically, it's a eukaryotic animal cell. Alright, animal cells are quite a simpler version of the eukaryotic cell in a way, because you don't have as many different components to it as a plant cell, but most of the major important stuff that we want to look at is in an animal cell. Let's start in the middle. Here you've got your genetic material. All right. For animal cells, this is going to be DNA. Right. When they're replicating, we do call that the chromosomes, though. All right. And where this genetic material is stored is in this membrane-bound organelle, which we call the nucleus. The nucleus's function is simply to protect the genetic material. All right. And then just outside of the nucleus, we've got this blue ribbed things, which we call the endoplasmic reticulum. All right, and you've got two types of endoplasmic reticulum. Mostly we talk about rough endoplasmic reticulum, but you can actually also have smooth endoplasmic reticulum. And what's the difference is that rough endoplasmic reticulum is surrounded by what we call ribosomes. And ribosomes are an extremely important organelle, which is pretty much essential in the manufacturing of protein. So when it's rough endoplasmic reticulum, you're talking about protein manufacture. and smooth endoplasmic reticulum, you're actually talking about lipid or fat manufacture. Right? These ribosomes can actually be found not only on the endoplasmic reticulum, but also they can be found just floating around in the cytoplasm of the cell. By the way, all of this yellow area is the cytoplasm. Okay, and then we've got this slightly darker blue organelle here, and that's called the Golgi, or Golgi, and I like to call it Golgi body, but you will see it being called Golgi apparatus in places. I just like calling it Golgi body because Golgi apparatus is harder to say. And then we have these tiny little clear areas which are vacuoles. Like I said, vacuoles are more common for plant cells, but they can be present in plant cells. All right. This purple body here, little purple organelle, is called the centriole, and this is going to be more important later on when we talk about cell division. All right. And then we also have very much the most important organelle, I would say, taking up the nucleus, is the mitochondria. Alright, where cellular respiration takes place, where the cell really breaks down sugars to make some energy. Okay, what else is there in here? We've got one of these that's likely a lysosome. Which is pretty much just a little packaging organelle. I think for most part, that's what we've got for a eukaryotic animal cell. If we were looking at a eukaryotic animal cell, you'd see something like this, right? Here we've got some eukaryotic blood cells. All right, they're red blood cells. All right, and then over here we've got another kind of blood cell, but a white blood cell. More specifically, it's a neutrophil. All right. Over here we've got some other eukaryotic cells. They're phagocytes. Oops, they're phagocytes. And then this is a dendritic cell. Different kind of, actually, they're all blood cells. But as you can see in the light microscope, you get really clearly a nucleus. All right, the red blood cells here don't actually. It's one of the few human cells that don't have a nucleus. But here you can actually even make out some other organelles there, likely mitochondria there. But yeah, not way too much information. On an electron micrograph, though, we get a lot more information, but we don't get color. All right, really clear nucleus, these centrioles here, you can see the plasma membrane itself. You can actually see, you can't see the plasma membrane, you can just sort of see the end of the cell when it's a light microscope. Really clear mitochondria, you can even see the inner membranes of the mitochondria. Pretty cool, the electron micrographs. Eukaryotic plant cell, we're actually talking about very much the same things. We have the nucleus, endoplasmic reticulum, both rough and smooth, the Golgi body, mitochondria. But some things that are different, right, are here we've got a very large vacuole. And that's really important because it fills up with water and makes sure that the plant cells are nice and turgid, nice and stiff, so that plants do stand up nice and straight. All right. We've also got a very thick cell wall. Right. And then probably the most important difference between eukaryotic animal cells and eukaryotic plant cells are these guys here, which are the chloroplasts. And they're going to be extremely important when we're talking about photosynthesis. All right, but as you can see, not too many differences between animal and plant cells. Okay, one thing that we couldn't see in that other picture of an animal cell that we can see here in the plant cell is this here, which is present in both, but that's the cytoskeleton of a cell. All right, cyto, if you ever see the word cyto, it means cell. So it's literally the cell's skeleton. It's the reason, it's how cells actually move around in this. All right, so under a microscope, I think plant cells look even cooler than animal cells, and that's one of the reasons we're going to be looking at mostly plant cells. But here we have some onion cells, all right? And then over here, we've got leaf, epidermal cells, all right? So when we look at cells under a microscope on Tuesday, you're going to actually see both cells of these both kinds. We're going to actually stain our cells so we do see really clear nuclei like this. But most importantly, and what I think is the coolest, is we're going to see cells, that's one individual cell there, that are filled with these green structures, which are called those chloroplasts. Under an electron micrograph, once again, we're going to get a lot more detail, but no color. All right, we're going to get chloroplasts that look like this, that you can see all the thylakoid membranes and such, which is really cool. But once again, just different kind of information that the electron micrograph is going to give us. Make sure you answer the 3 quick questions, and I'll see you guys in class.